Hi everyone. So in the previous lecture we studied about the structure of the oral mucosa and we saw that it had these layers: the epithelium, the basement membrane, the lamina propria and the submucosa. So we studied about the basement membrane, lamina propria and submucosa in somewhat detail, whatever was required. Now in today's lecture we'll cover the structure of the oral epithelium. So this layer we'll study in detail today. So the structure of the oral epithelium, it is made up of stratified squamous cells, okay? And the epithelium, it can be either keratinized or non-keratinized. So based on the keratinization, it is classified into keratinized epithelium and non-keratinized epithelium. The keratinized epithelium, it includes the gingiva and the hard palate and vermilion border of lip. The gingiva and hard palate, they together, it is called the masticatory mucosa because it is involved in mastication or chewing, right? Or and the non-keratinized mucosa, it includes the lining mucosa and specialized mucosa. So specialized mucosa is the tongue and your taste buds. And lining mucosa is everything other than the keratinized and the specialized mucosa. So whatever remains in the mouth, all this buccal and floor of mouth, this is everything is lining mucosa. Okay. Now we'll first start with the structure of the keratinized epithelium and then we'll see about non-keratinized epithelium. Okay. So looking at the keratinized epithelium, I'll tell you the basic construct of the keratinized epithelium. It has four layers, okay? So it has one, two, three, four layers, okay? These four layers, they are first consist of some keratinocytes, these cells, and other than these cells, it also has some non-keratinocytes which will lie all over the epithelium, okay? The keratinocytes, they will produce keratin as the name suggests, and the non-keratinocytes, they have different functions. We'll study that in detail. So first we'll study the process of keratinization, okay? How these keratinocytes are formed. This is the process of keratinization. Now if I start with the first layer, so I told you it has four layers, right? So the first layer, it is called the basal layer. The second layer is called the spinous layer. The third layer is called the granular layer, and the fourth layer is called the stratum corneum. Okay, so the basal layer. Now, what will happen that these cells, the cells will divide from the basal layer, and they'll keep moving upwards, and they will get biochemical changes. They'll have some biochemical changes, and ultimately, the basal layer cells, they will transform into stratum corneum. So how that happens, this entire process is keratinization. So I have this cell of a basal layer. Okay, it will undergo mitosis. It will form one cell that will stay in this layer and the second cell that will go upwards. Okay, now it is a cell of the stratum spinosum, right? It has some biochemical changes, some changes happened in here. Now it will move upwards and it will become a part of the stratum granulosum. Okay, again some biochemical changes will happen in here, and now ultimately it will become a part of the stratum corneum, the top layer. What will happen in the stratum corneum? The nucleus will die off, the cell organelles will disappear, the cell will basically become dead. The dead cell, it will be called a squama, and then it will be shed off. Okay, so the shedding off, it is called desquamatization. Okay, this is the entire process of keratinization. Right. So now we'll just look at the notes. So keratinization, it can be either ortho or parakeratinization. Right. I'll tell you what the difference is between ortho and para in later on. It consists of four layers. So the basal, spinous, granular, and stratum cornified layer. Okay, these are the keratinocytes and some non-keratinocytes, as I told you. Mitosis occurs in the basal layer. After that, the cells migrate and are pushed upwards and they undergo differentiation. So, this process of differentiation, I told you that the basal cells will undergo mitosis and they will move upwards, and some biochemical changes will take place. Those biochemical changes, that is differentiation. and this entire process of differentiation, it is called maturation. Okay, time taken for maturation, it is called the turnover rate. Non-keratinized epithelium is faster than the keratinized epithelium. Okay, differentiation, it will end with a formation of squama, a dead cell, and shedding off. Okay, so the squama will shed off. That's how this keratinization will end. The cells are shed off, that is called desquamatization. The whole process is keratinization, right? Now moving on to keratinocytes and non-keratinocytes. So some details about that. So keratinocytes, these are the cells which synthesize keratin, okay, and they have a characteristic filament protein called cytokeratin, right? Non-keratinocytes, they are smaller in population, okay, they are less in number. They do not have cytokeratin, okay, and they do not keratinize. Uh, so they have different functions. They are not for keratinization. They do not show mitotic activity and they do not undergo this differentiation or desquamatization. Okay. Now looking at this protein cytokeratin, there is some detail given in the book. So cytokeratin, it forms the cytoskeleton of all epithelial cells. Okay, so it forms the cytoskeleton. It provides mechanical linkage. Okay, and it distributes the forces over a wide area. So this is what the function of cytokeratin is, and it helps in maintaining cell shape. So this is the function of cytokeratin, and the main thing is that it is present only in the keratinocytes. It is absent in the non-keratinocytes. So remember that. Now moving on to the keratinocytes, these cells, okay, what happens is, so looking at the first layer, the basal stratum basale, it is a single layer of cuboidal cells. Okay, they synthesize DNA and undergo mitosis. So only this layer will undergo mitosis and new cells are generated. The cells become determined as they leave the basal cell. So what it means by determination, determined is that they are ready to differentiate. Okay, so as the cells leave the basal layer, they are now ready to differentiate. Okay, stratum spinosum, the second layer, it is also called prickle cell layer. So the cells will look somewhat like this. Okay, they will have a spinous like kaante wala appearance. Okay, they are irregularly polyhedral in shape. So the cells are polyhedral. They are larger than the basal cells, so the cell size will keep on increasing as we go up. Okay, so these ones are larger than the bottom layer, the basal cells. They are connected by desmosomes and tonofibrils. So desmosomes are like linkages between the cells, and tonofibrils are these red ones which will go across the cells. Okay, and these together, the desmosomes and tonofibrils, they will form a tensile supporting system for the epithelium. So basically, it will provide the strength to the epithelium. The spiny appearance is due to shrinkage of the cells during tissue preparation at points where desmosomes are not present. So maybe this cell was like this at one point of time, and then we did tissue preparation. So the entire cell shrunk. Okay, it shrink, and then at this point it was held by a desmosome. So at this point stayed over here, but shrinkage occurred over here. Okay, then again this was held by a desmosome, so it stayed, and then it got shrunk over here. So because of this shrinkage, the cell had this spiny appearance. Okay, and that's why it is called stratum spinosum. It is the most active layer in protein synthesis. So remember that the first layer, it has DNA synthesis and it has mitosis, but this is the most active layer in protein synthesis. Now we'll look at the third layer, the stratum granulosum over here, right? So this one over here. The cells are wider and larger than the spinous layer. So again, the size is increasing, and now they are getting wider. Okay, so now they're not increasing in height, they are getting wider. It is basophilic, so it stains blue with H&E stain. The nuclei show sign of degeneration and kenosis, and they still synthesize proteins. So I told you that in the top layer, the top layer will be a dead layer. Okay, so now in the granular layer, the nuclei has started dying. Okay, so now it's showing signs of degeneration, in here, but it is still alive. It still synthesizes proteins. An organelle called keratinosome or Orlando body can be seen in the upper spinous and granular layer, which releases their content into intercellular space, forming a barrier at the junction of granular and cornified layers. So what it means is, if I go in over here, let me just erase this. So in the granular layer, I have an organelle called Orlando bodies. Okay, so these are Orlando bodies. What will happen? These organelles, they release something into this. Just a second, I have to shorten this. And they will release some substance into this intercellular spaces. Okay, and this substance, it will form a barrier between the cornified layer and the granular layer, and this will affect the permeability of the overall oral mucosa. Okay, so this is the significance of the Orlando bodies or keratinosome. Now coming to the top layer, the stratum corneum, the cells are larger and flatter, so it will have the largest cells. And now all the organelles have disappeared. So I told you there are two types of keratinization, right? So there's orthokeratinization and there is parakeratinization. So you will see the difference in this layer. In orthokeratinization, I will have no nucleus, nothing. Okay, it will be an empty cell with proteins. But in parakeratinization, I will have some remains of a nuclei, right, and organelles, and when until the cell is shed off. So this is what these lines are saying, that in orthokeratinization there is no nuclei in this layer, but in parakeratinization we can see some remains of nuclei and uh, organelles until the cells are shed off in this layer. The layer is acidophilic, so it stains red with H&E stain. The cross linking, okay, so this point, what it means is that this layer has disulfide bonds, which basically provides mechanical and chemical resistance to this layer. Okay, just a second. Now coming to the non-keratinocytes. So looking back at this diagram, uh, yeah, this one, I told you that we had these keratinocytes, these cells. Now we'll look at the pink dots. Okay, these are the non-keratinocytes. They do not produce keratin. They don't have the mechanical functions. They have different functions, and we'll have to study that. Okay, so looking at the non-keratinocytes, if you look at this diagram, so this is the structure of, this is the oral epithelium. We had the first layer basale, the second layer spinosum, the third layer granulosum, and the fourth layer cornified. Okay, we have these cells. These are the non-keratinocytes. They are dendritic mostly, and they have different functions to keratinization. They do not produce keratin, and we'll have to study their functions. Okay, so these are usually dendritic, and they appear unstained or unclear in H&E stain. Okay, uh, basically there are three non-keratinocytes. There is Merkel cells, Langerhans cells, and melanocytes. Okay, there are other inflammatory cells like lymphocytes. Those are also included in non-keratinocytes, but we don't study them. We only study these three. Okay, and they generally originate from neural crest cells or bone marrow. So that is where they come from. Okay, this is very important also. So the first cell we study is melanocytes. We know that melanocytes, they produce melanin. So they are present in the basal layer. So you can see over here the melanocyte is present. It is present in the basal layer. They generate melanin. They originate from neural crest cells. What are neural crest cells? You study that in embryology. So, but basically there are some cells that are formed during neurulation. Okay, so remember that. And so that's where these cells come from. They transfer melanin through dendritic processes to keratinocytes, which store melanin in the form of melanosome. So these cells, they will make melanin. They will transfer it to the keratinocytes, and the keratinocytes will store the melanin in the form of melanosome. Okay. Now these are also called clear cells or dendritic cells. Clear because they are clear or as in transparent in H&E stain, and they are called dendritic cells because they show their dendritic appearance in silver stain. So they can be seen through silver stain. Remember that. Now the second, that is the Langerhans cells. So you can see over here, these are the Langerhans cells. Okay. They are also dendritic in shape. They originate from the bone marrow, and they have convoluted nucleus and some granules that are called Birbeck granules. Okay. So these are dendritic in shape, like other non-keratinocytes, originate from bone marrow. They have a convoluted nucleus and some Birbeck granules. Okay. They are stained with gold chloride, ATPase, and immunofluorescent markers, and they are involved in the immune response. So the main points you should remember for your university examination is these are also dendritic, they originate from bone marrow, and they are involved in the immune response. Okay. The third one, the Merkel cells, over here, right? The Merkel cells, they are also present in the basal layer. They have nerve tissue, which is specialized pressure sensitive receptor cell. So they basically respond to touch. Okay, these cells, they respond to touch. They have receptor cells. These are non-dendritic, so very important to remember, because generally non-keratinocytes, all non-keratinocytes are dendritic other than Merkel cells. So these are non-dendritic. They also originate from neural crest cells, so like the first one, like the melanocytes, and they are stained by pastain. Okay. Now we saw, so going back to the first slide, we studied the structure of the keratinized cell, keratinized epithelium. We saw how keratinization takes place and everything about the keratinized epithelium. Now we'll look at the non-keratinized epithelium. Okay, so the template will remain the same, but the non-keratinized epithelium, it has some differences to the keratinized epithelium, and we'll see what the differences are. Okay, so first of all, the mm, what's happening? Okay. Uh, so first of all, the non-keratinized epithelium, it had four layers. Okay, it had four layers we saw. It had the basale, it had spinosum, it had granulosum, and it had the cornified layer. The non-keratinized epithelium, it will only have three layers. Okay, so it will have the first layer basale as normal, the second layer is intermedium, and the third layer, it is spinosum. Okay, so it has only three layers. So we don't have we don't have any cornified layer, or we don't have any granulosum in the non-keratinized. Okay, so this is what it says. It does not produce cornified surface layers. There are three layers: basale, intermediate, and superficial. Sorry, not spinosum. I wrote spinosum. The top layer is called the superficial layer. Okay, so it has three layers. Right. The cells of stratum intermedium are larger than the cells of stratum spinosum. So if we compare the second layer of both the keratinized and non-keratinized, the intermedium one, it will have larger cells. Okay. The cells do not have a prickly appearance. So the spinosum had a prickly appearance, but the intermedium won't. So it does not have a prickly appearance. There is no stratum granulosum. So the granulosum is absent, and this has a higher rate of mitosis. So in the turnover rate, we studied that the non-keratinized epithelium is faster, so it has a higher rate of mitosis. Right, so these are the differences between keratinized and non-keratinized epithelium in the structure. Now I'll just give you a brief review of what we just studied. So we had our oral mucosa. It had these layers: epithelium, the lamina propria, the submucosa, and it had a basement membrane, right? So this was the oral mucosa. Now we went into detail about epithelium. It was of two types: it was keratinized and non-keratinized, right? The keratinized epithelium, it consisted of keratinocytes and non-keratinocytes, right? So we had four layers. One, two, three, four. Okay. In the keratinocytes, we had basale, we had spinosum, we had granulosum, and we had a layer of cornified cells. Okay. And this, the process of formation of this entire thing was called keratinization, where the basale divided and it formed all the following layers, and then ultimately it was shed off. Then we had non-keratinocytes in between these layers, where um, we had melanocytes and Langerhans cells in the basal layer, and then we had Merkel cells in the above layers. Sorry, we had Merkel cells and the melanocytes in the basal layer, and we had Langerhans cells throughout the layer. Non-keratinocytes, they had different function. So melanocytes, they produced melanin. Langerhans cells, they had an immunity function, and then Merkel cells, they had a receptor function. Okay, we studied that. Now in the cornified layer, it was ortho or parakeratinized, right? The orthokeratinized, it had no nuclei, nothing, but the parakeratinized, it had some nuclei, some remains in there, right? The the spinosum layer, it had a prickly appearance because of tissue preparation, right? In the non-keratinocytes, we had only three layers. So we had basale, we had intermedium, and we had superficial layer. The main differences we saw was, so there's no cornified layer in here, there's no granulosum layer. uh, if we compare the spinous layer, if we compare the spinous layer and the intermedium layer, so the intermedium layer is bigger and it does not have any prickly appearance. Okay, so this is basically all what we studied in here, uh, in this lecture, and in the next lecture I'll cover masticatory mucosa. We'll go through that in detail in the next lecture, and if you like this lecture, please like and